Hi everyone, welcome to
amazing channel of Brilliant Katha.
When you're rubbing one body with another body, electrons are transferred from one object to what? Another object. Okay, this process, this type of electricity is been given a name.
halogen connected carbon sp3, then it's connected to benzene. See all of you, halogen connected. What is this carbon hybridization? Full single bond sp3, see then a benzene ring.
Denominator poles shouldn't be included in the denominator poles. That means the denominator, these -1 and -5 should never be included in the solution.
Homocircle caudal fin. It's giving two equal lobes. But in case of cartilage fishes, look at here, one big lobe is there, one small lobe is there. You cannot cut into two equal half this fin.
Brilliant Katha.
Hello students, good evening you all. Welcome to Brilliant Qatar. Now, Masters NEET. This is a time for preparation for NEET and also what? Biology CBSE. I'm a clear student. So far, physics done, chemistry exam also done, mathematics also finished. So next exam is what? Biology only. What sir?
Yes, biology exam will be happening soon. So we are going to start with some NEET preparation questions also, along with some topics that will be very important in the coming board exam. So we will be conducting the molecular basis and principles of inheritance chapter. These chapters are having a good weightage for the board as well as NEET. Sir, how many questions you are expecting for NEET and all?
In NEET examination, principles of inheritance 2 to 3. Molecular base of inheritance 3 to 4 questions can come, and for the CBSE examination also these chapters are very important. And if you are able to recall when we are discussing theory classes, this unit is how much weightage? 20 marks in CBSE. Do you remember? Evolution, principles and molecular.
So it's having a good weightage in the coming exams and all, so you should be very aware when you are preparing.
Yes. So my dear students, whoever lagging these two chapters, you don't know monohybrid and dihybrid cross or a mutational theories or mutation or pedigree, this is the right time, this is the right platform to cover up. So I'm requesting you all take pen and paper and try to understand the questions and try to write down the concepts which we are going to discuss.
So sir will be starting with the most important questions coming in molecular basis as well as principles of inheritance. So we'll be discussing all the concepts that are coming along with the MCQs and all. So please stay and watch the live.
Right.
Okay.
Are you ready, students? Shall we start? Look at the first question.
Yes.
So the first question from principles of inheritance and variations. So these questions are NEET level based question and also these are competitive based questions. Student, this will be utilized for the CBSE purpose also. Okay. So look at the first question. The first question is given below is highly simplified representation of the human sex chromosomes is given, and from a karyotype. We know the karyotype, but a karyotype is the entire the chromosomal map, right? Karyotype. And the gene A and B could be of the given. They're given gene A and B, the two chromosomes are given. Sir, in the question they mentioned sex chromosome, but which one is X chromosome, which one is Y chromosome, we don't know. Remember students, if you're able to recall the theory classes, the X chromosome is bigger than the Y chromosome. X chromosome is bigger than the Y chromosome. These are actually a sex chromosome. Sex chromosomes are not homologous. They are not complementary. Do you remember? This is a male condition, right? So in the male condition, what happened? This one is X chromosome and the small one is called what, beta? Y chromosome. So when XY chromosomes are two different, we call them male. You got my point? That means, so can any one of you tell me gene A and gene B are located on which chromosome, my dear? X chromosome. So that means they're talking about the sex-linked recessive disorders. or X chromosomal disorders? Find out the answer. Look at the question. Color blindness. Yes, color blindness is a sex-linked recessive disorder. What about the body height? Do you remember body height, body color? These all are autosomal. So the genes are located on autosomes. Ignore this option. Leave it this option. Am I clear? Look at the second option. Attached earlobe is autosomal character, my dear. It is not a sex-linked character. So leave it this. Am I clear? And rhesus blood group. Do you know the rhesus blood group? RH they are asking about. RH positive, RH negative, capital D, small d, we discussed in 11th grade bio body fluid and circulation chapter. This is the RH antigen they're talking about. Leave it. Now hemophilia, right? Hemophilia is what type of disorder? Tell me. It is a sex-linked recessive. Fine. And also color blindness. Yes, color blindness also located on where, my dear? X chromosome only, right? So these are the sex-linked recessive. If you're able to recall how many point mutation, 6 type of point mutation we discussed in the theory classes, right? Out of which 4 are autosomal recessive. What are they? Phenylketonuria, sickle cell anemia, thalassemia, and also what? Cystic fibrosis. These all are autosomal recessive, and 2 sex recessive also we discussed. Can any one of you tell me what are they? Sir, colorblind and also hemophilia. So look at the hemophilia and also colorblind. They given. Fine. This is the appropriate option C. And look at the option D. What's wrong in option D? Phenylketonuria and hemophilia. Sir, hemophilia sex linked, but phenylketonuria is what, my dear? Autosomal. Am I clear? Autosomal. And one more important thing, the genes which are located in chromosome, Y chromosome, what kind of genes are there? These are called Hollandric genes. Remember, Hollandric genes, but they did not mention any gene on Y chromosome. So what is our correct answer? What is your correct answer? Any one of you? Any one of you give me the right answer? The X chromosomal genes are hemophilia and also color blindness. So our first question, diagrammatic question answer is option C. Don't get panic if this type of questions come. Think they're given a sex chromosome, X and Y chromosome, and what they are asking. Simple answer, my dear. So first question answer is what? C is the first question answer. Second question, right, sir? Katie, sir, come.
So one more question. Please comment. Who discovered X chromosome and who discovered Y chromosome?
Simple, simple.
Who is going to comment it first?
Simple answer. Who
So,
discovered?
sir,
Who discovered X chromosome and who
discovered
given the Y chromosome?
So we are going to check the comments. Who is going to make
fast,
it first?
fast.
So let's see.
Any one of you?
Anyone of you, please comment fast. Yes.
Yes.
Reem has commented.
Very
Yes.
good. Absolutely
So
correct.
what is the answer?
What is the answer? Come on.
Henking only discovered X chromosome. What about Y?
Henking. Y chromosome. Any one of you?
Chromosome. Who is going to answer? Reem got the first answer. Let's
Right,
see who
good,
is going
very
to get
good.
the second one.
Any one of you?
So, okay, Magasri also commented Henking is the answer for
Yes,
X
yes,
chromosome.
correct,
What
my dear.
about Y
Y
chromosome? We just
chromosome.
want to
Can
know
you-
who discovered Y chromosome?
Any one of you? Hollandric genes, Y chromosome. Fast.
Stevens, the scientist actually who discovered it. is
Steven
Stevens.
Wilson. Right, good, very good. Come.
Okay, so now
Next
let's
question.
start with the next question. Okay, sir.
Yes, continue.
So we'll be starting with the next question from Principles of Inheritance. Among the 7 pairs of contrasting traits in a pea plant, as studied by Mendel, the number of traits related to flower, pod, and seed respectively were. So how many characters are coming from flower? How many characters are coming from pod? How many characters are coming from seed? You know 7 pairs of characters he was learning. In that, the flower he was actually learning 2 characters. One is flower color and flower position. In that, dominant is the violet and recessive is white. And flower position, if you take axial and terminal, is the dominant and recessive. So 2 characters he was learning from flower. And pod, if you are taking pod color and pod shape. Pod shape, you can see it is inflated or full. It is full or constricted, like that 2 characters. Then he was also learning about the pod color, that is green and yellow. The dominant is green, recessive is yellow. Then seed, also 2 characters were learned, that is the seed shape as well as the seed color. In seed shape, you can see it will be having round as dominant, wrinkled as recessive. But the color is just opposite. The dominant is yellow and the recessive is green. So all of them, yes, very good. Oh, already answered. Kridya, that is the answer. It is 2, 2, 2. The answer is 1. It is 2, 2, 2. So hope you know the 7 pairs of contrasting trait. And Mendel was taking around 14 true breeding plants for learning these 7 pairs of contrasting characters. Now in that, 1 character only I have left out here. Can you comment which character? So we discussed about flower color, flower position, pod color, as well as the pod shape, seed color, as well as the seed shape. One more is coming. Which one is remaining? Can you please comment first? Which one? Which is the seventh character? That is the first one, stem height. Yes, very good. Dia commented it is stem height, tall and dwarf. Like this, 7 pairs of contrasting characters was led by Gregor Johann Mendel. Now let's move to the next question. Next question will be from Sandeep sir. Yes.
Yes, sir. Now look at here. This is also one of small, simple question. Look at the question. I hope many of you got the answer of this question. Simple question. Both parents are carrier for thalassemia. First, answer me. Thalassemia is a sex-linked or recessive? Sex-linked or recessive? Thalassemia, beta thalassemia, alpha thalassemia, we know this one. Thalassemia is autosomal recessive, right? It is not a sex-linked, leave it. Now, which is autosomal recessive? Oh my God, already in the question the given, my dear, it is autosomal recessive disorder. What are the chances of progeny resulting in affected child, the given? So, remember, if it is autosomal recessive, I can simply take as a capital A, small a, both parents are carrier, right? They given a carrier. Both parents are carrier. That means they are not showing any disease. No thalassemia for parents. Now tell me here. Simple answer. Now here the 2 type of gametes can come. Capital A, small a. Here also capital A, small a. Tell me when will this disease express? When 2 recessive genes present only will express, right? Because it is autosomal recessive. Recessive condition will be only this genotype only will express. So what about capital A, capital A? Both dominant genes are there. They will say, no, I am here, the dominant gene, how can you express? So there is no chance of here. What about here, capital A, small a? This condition also capital A said, I am here, how can you express? So the dominant, it will not express. In this both condition, they will not express, my dear. Tell me, capital A, no chance. Beta, capital A, small a, no chance. Capital A, small a, no chance. Last one is what? Tell me, small a, small a. So what percentage? Can you look at the now shall I like 25%. So simple answer, the right answer is option B. Don't get confused in the exam. Simple. Try to think and try to read the question once and twice before your answer. You got my point? Shall we go to the next question? Look at the next question.
Before that, one more question.
One more question.
Which type of inheritance it is? Is it qualitative or is it quantitative?
Yes. Good question. Is it qualitative or quantitative? Now, thalassemia
is an example of qualitative or quantitative.
Yes, nice question. Any one of you. Come on, fast.
Who is going to get it first?
What kind of qualitative, quantity? Quantity, quality. Think, my dear. Quantity, quality. Come on.
First answer.
Yes. Reem is rocking today like anything. Very good, beta. In sickle cell anemia, problem in the hemoglobin manufacture. So it's called what? Tell me. Qualitative. You got my point, but this is quantitative. Remember, all of you. Got my point? Look at the next question. Come, sir.
Yes, genes which code for a pair of contrasting traits are known as. You already know about it. But still, you have to keep in mind, in a pair of chromosome, which is called as homologous chromosome, at a locus, different genes are coming. For example, at this locus, if stem height gene is coming, it can have 2 possibilities. Either it can have tall or it can have dwarf. Here also, it can have either tall or it can have dwarf. So those chromosomes which are carrying similar genes in a chromosome, or similar characters genes in the chromosome, they are called as homologous chromosome. So this alternative form of a gene or a trait that is coming at same locus of the homologous chromosome is called as what? They are called as alleles. So hope you got it. They are alleles. Okay, all of them were commenting first of all. So the answer is that only. Now let's move to the next question. So please remember the explanation. Come sir.
Right. Look at here. Look at this question. This is a little lengthy question, right? Look at here. The question tell us human skin color is controlled by several gene pair. The given human skin color is the example of polygenetic inheritance, quantitative inheritance. Do you remember this one, my dear? Tell me here what happened. Polygenic inheritance. Polygenic inheritance is that inheritance that means one trait is controlled by many or several, one trait controlled by several genes. You got my point? Many genes are there. What are they? Capital A, capital A, capital B, capital B, capital C, capital C. 6 dominant, all dominant. What is the skin color? Very, very dark skin color. So what about this condition when recessive small a, small b, small b, small c, small b, small c, albino? It is the albino autosomal recessive condition. So what happen if both are married here? Now I can get that capital A, capital B, capital C type of gametes, and here it will be small a, small b, small c. You got my point? Fine. What they given? Human skin color is controlled by several gene pairs. Let us assume here that there are just 3 gene pairs on different chromosomes, and for each pair there are 2 alleles on incompletely dominated, one on code for the melanin deposition, and incompletely recessive one that code for no melanin deposition. Fine. If a very dark skin color person marry a very light skinned woman, capital A, capital A, capital B, capital B, capital C, small a, all are small a, what will be the chance of their offsprings will have very dark skin color? Krishna sir, can you cross check? Anyone give an answer? Anyone give an answer? What is given? Yes. Look at here. In this condition, when we cross this one, what we will get? Tell me, capital A, small a, capital B, small b, capital C, small c. My dear, tell me what kind of this one? Tell me this is intermediate, right? Yes, sir. Is it intermediate or not? Intermediate or not? Yes, tell me. So what will yes. So answer is what? Tell me. What is the answer of this one? Anything is there? Yeah, no, right? Tell me. You got my point, my dear.
Okay, first comment was from Dia.
Yes.
Commented first, so Dia only commented first. Dia's answer.
And moreover, my dear kids, even you're preparing this question, remember the phenotype ratio. 1:6 something I given, right? That phenotype ratio, remember, if these two, capital A, small a, capital B, small b, capital C, small c, cross with same capital A, small a, capital B, small b, capital C, small c, we will get what? Tell me, 64 out of 64 the phenotype ratio I given, cross check. But I have a small question. Krishna sir, cross check the comments. How many different type of gametes can come from this genotype?
How many gametes are possible?
Different type of
genotype. How many different gametes are possible?
Can any one of you from this genotype?
This one. Capital A, small a, capital B, small b, capital C, small c. How many? How many is possible?
How many different? How many different type of gametes?
8.
Yes.
So what is the equation?
Very good, very good.
For finding the number of gametes.
2
to the power
n. Simple.
But n you can use as the number of
heterozygous.
So how many? 3 heterozygous.
3 heterozygous. So 8. Remember. You got my point? Go to the next question.
So here, next question. What will be the distribution of phenotypic features in the first filial generation after a cross between homozygous female and heterozygous male for a single locus? Here homozygous female they have mentioned about recessive only. So if it is a recessive homozygous female and a heterozygous male, then what can be the ratio? Actually they are telling about the test cross. So if I am taking a homozygous female and a heterozygous male, like this, a homozygous female and a heterozygous male, you know what cross it is? It's actually the test cross, where you will be getting which ratio? Where you will be getting, this one will be tall and this one will be dwarf. So you will be getting 50% dominant and 50% recessive. So that is an example of test cross. The ratio is 1:1. So remember, the monohybrid test cross ratio is 1:1. Also, please comment what is the dihybrid test cross ratio. Let's see who is going to comment this.
Dihybrid. Can any one of you? Dihybrid
test cross ratio.
Dihybrid test cross ratio.
Who is going to comment first?
Any one of you.
Anyone. Many has given the 1:1 here. So we are expecting for the dihybrid test cross ratio. when F1 is crossed with the recessive parent.
That means let me give small hint. Capital T, small t,
Kavya,
capital
Kavya
R,
has
small
already
r.
commented.
Yes, very good, very
Kavya
good.
got the first answer. Kavya, it's the answer.
Yes, Sabas, absolutely.
1 is to 1 is to 1. Sir, please show that dihybrid test cross also.
Right. Okay, look at here.
So just look at
All of you look at
showing dihybrid test cross, crossing the F1 with the
Capital R.
recessive parent.
Yes, capital R, small r, capital T, small t. This I am taking. Okay, this is a F1, right? These are the F1.
F1.
Right. I am recessive parent. What's the recessive parent? Tell me small r, small t, small t. So tell me here how many different type of gametes can come? Capital R, capital T, capital R, small t, small r, capital T, small r, small t. From this genotype. I am a clear student. From here tell me small r, small t, small r, small t, small r, small t, small r, small t. Fine, correct. Now tell me here what is the cross of this one? Tell me capital R, small r, capital T, small t. So this all are 1:2. Am I clear? You got my point. All are same, right? Next, what about this one? Tell me capital R, small t, small t. 1, 2, 3. So 1:2. 1:2. Now look at the third one. Tell me small r, capital T, small t. 1. Last one is tell me small r, small t, small t. So 1:2, 1:2, 1:1. Simple.
So if you take the phenotypic ratio, you will be getting 25% round tall, 25% round dwarf, 25% wrinkle tall, 25% wrinkle dwarf. So it will be 1:1:1:1.
Simple.
That is the dihybrid test cross ratio. Remember that. Now sir will be starting with the next question. Yes sir, come on.
Look at here. The next question. Next question. Any one of you? By the looking of this question, within 30 seconds we should get the answer. Come on, fast. Which of the following is a sex-linked disorder is given? Sex-linked disorder. In pre-board 2, I given same question. Many of students are getting confusion between the sex-linked disorders and also what chromosomal disorders. Look at here what is given. Which of the following is a sex-linked disorder? Sickle cell anemia, autosomal. Albinism, autosomal. Albinism means what? Small a, small b, small c, small c. Last, hemophilia. I don't know, you have to cross check. Phenylketonuria, autosomal. So answer is what? Answer fast. Very good. Simple. And one more important thing, my dear. Chromosomal disorder to point mutation. This point mutation difference is there. In case of point mutation, number of chromosomes are same, no change. Let me take a small example. For example, if I am taking sickle cell anemia, 46 chromosomes are there, beta. There is no change in the chromosome. It is a point mutation, single nucleotide base pair alteration. But in case of Down, Turner, Klinefelter, what happened? Number of chromosomes will get changed, aneuploidy. You got my point? So the right answer is what option? C. Simple. You got my point?
Already they
very good. Go to the next question.
Now next question, if 4 o'clock plants, the gene for red flower color, capital R, is incompletely dominant over the gene for white color, small r. Hence the plants are heterozygous for the color, capital R and small r, and pink flowers are produced. You know already this is an example of incomplete dominance. So here the question is, what will be the ratio of offspring if cross between the flowers, red flower it is capital R. If it is crossed with the pink flower, that is capital R, small r. Here it will produce only one gamete, here it can produce 2 gametes. Yes or no? So here you will be getting 50% you will be getting which one?
Good.
Red, and 50% you will be getting pink. So what is the answer? 50% red flower and 50% pink flower.
Tell me the example of this incomplete dominance.
Another example, please keep commenting. What are the other examples?
What is given here?
Incomplete dominance?
It is 4 o'clock plant is given,
Other
right?
than 4 o'clock
Anyone,
plant, which all examples you know, keep commenting.
Come on. Any one of you
who is getting it?
Yes, Faiza. What is the answer of examples of incomplete dominance?
Incomplete dominance. Okay, Snapdragon.
Yes, very good. Absolutely. Next, Andalusian fowl. Do you remember? Andalusian fowl in case of birds. Remember
antirrhinum. Okay,
very good. Sabas, very good.
Okay,
next,
sir. Next question.
Come to the next question. This is take time, my dear. You need to be very careful. I'm requesting you all. Sex determination, thoroughly prepare. Sex determination, male heterogamety, female heterogamety, thoroughly prepare and go. This academic year, this time there is a high chance from chromosomal and also what they call sex determination. In male grasshopper, first you need to understand grasshopper shows what type of sex determination. XX and XO type of sex determination. This is actually male heterogamity. What exactly meaning of this male heterogamity? Male partner is going to produce two different type of sperms. Look at here, two X chromosomes are there in female, only one X chromosome is there in the sperm of grasshopper. But look at the question. Grasshopper and moths, there are two pairs of autosomes are there and one. What one among this one? Only X, only Y, XY, XX. Can any one of you answer? Any one of you answer? What is the correct answer? In male, what is there? Sir, in male, autosomes plus only one X chromosome. Second one is not there, right? It is in the male. In case of female, what my dear? A autosome plus both X chromosomes are there, XX. But this is in case of male, only one X chromosome. So the right answer is what? Can you cross section? How many are given? How many are given? Absolutely, absolutely.
Yes.
So answer is what? Option A.
Very good, very good. Keep commenting.
This time sex determination, don't miss, my dear. Go to the next question, sir.
Yes. Next one. What can be the blood group of offspring when both parents are having AB blood group? You already know about doing the blood grouping and all. Remember, blood grouping is an example of multiple allelism. In multiple allelism, you should understand a single locus of a chromosome will be having more than two set of genes. That is possible. So here, there are three alleles that are coming for blood grouping, IA, IB or IO. Any of this can come at a locus. So it's an example of multiple allelism. You already know what is the genotype of this and all. So AB blood group. So here, if I take father that is having IA, IB, this is an example of codominance.
Yes.
Where you know that both the genes
will
are
express.
expressing. And female also you can see it is IA and IB. So when you are doing it, you just have to put it in the Punnett square and you have to do it. That will be
simple.
Good. So this will give you the right percentage and all. So male gametes you can see it can be either IA or IB, and the female gamete also you can see that can be either IA or IB. Like that you will be getting. So you know that already the four blood groups are available. IA, that will be A blood group, 25%. IA, IB, that will be
AB blood group.
IA, IB, that
AB
also will be AB blood group. So 50%, 2 out of 4 is AB blood group.
Yes.
And
IB,
25% will be B blood group. So here if you are taking, you can see they are having A, AB and B. So what is the answer?
A, AB and B.
I think many of them has already commented the answer. So some percentage wise the question is coming. It's better to do it in the Punnett square in order to avoid the mistake. So easily you can get 1 out of 4 is A blood group, 2 out of 4, 50% is AB blood group, and 1 out of 4 is B blood group, 25%. So hope you got it. Now let's move to the next question. Already comment has come. Very good.
Very good.
Yes.
Look at this question. Look at this question. Any one of you, try my dear. Try this. Look at here. This question is tell us normal vision man whose father was colorblind. We know, my dear kids, many times I said in the class, from father to son it will not transfer because this is a color blindness is sex linked recessive. From where to where it will go, tell me? Mother to son, father to daughter. You got my point? So here the normal vision man whose father was colorblind, that means this fellow is normal fellow, XY, normal person. Marry a woman whose father also colorblind. That means this woman, who is this woman? Can any one of you tell me the genotype of this woman? Any one of you tell me who is this woman? Who is this woman? Any one of you, who is this woman? Is this a carrier or affected person? Tell me, carrier or affected person. This fellow, this woman whose father colorblind, right? So I am writing here, she is normal, XC, X. She is a carrier. I am clear? She is a carrier. Chalo, I find out the answer. Find out the answer. Can you do this one? come on. Now from here two different type of XC, X, and here X and Y. Done. Now from here XC normal, and from here XCY affected male, XX normal female, XY normal male. Fine. They have first child as a daughter. What are the chances that child would be colorblind daughter? Look at here. This is a first daughter. This is a second daughter, right? There is a first daughter. It is a colorblind possibility. Come on, any one of you know? Yes, absolutely 0%. There is no, because carrier is there, and also what one is a normal. So answer is what? Absolutely correct. Absolutely simple question. Very good, my dear. Jalo, go to the next question.
Okay, already many comments are coming before we are discussing itself. Very good. Keep on commenting like that. Do the questions fast. ABO blood grouping in human beings sites, and it is actually the example of ABO blood group. If you are taking, it's an example of codominance and multiple allelism. In that, please do remember A blood group, B blood group, and O blood group. It is an example of complete dominance. It's an example of complete dominance. Only AB blood group.
Yes.
That is an example of
codominant. Both are expressing.
Yeah, it is an example of codominance, and it's also an example of multiple allelism, because just now we discussed at a locus of a chromosome,
3 alleles.
more than 2 set of alleles are coming. That is an example of multiple allelism. It's a question from 2018 NEET also. Please check that question paper, you will find it. ABO blood group, it's an example of multiple allelism, codominance, as well as it's an example of complete dominance in that A, B, and O. Okay, then let's go to the next question. Yes,
look at here. Most important, my dear kids. This time I told you sex determination thoroughly. You should prepare and go. Look at the question. Tell us, which of the following conditions correctly describe the manner of determining the sex in given example? Which of the following is the correctly determine the given? Four statements given. Out of one is correct. Out of 4, 1 is correct. Homozygous sex chromosomes ZZ determine the female birds. My dear kids, in female birds ZW, right? It is not ZZ. ZZ is male. ZW is female heterogamety. Do you remember? Now, look at that XO type of sex determination. XO type of sex determination is in male sex of grasshopper. Yes, before 2 minutes before I taught this one. Yes, absolutely. This is correct statement. What's wrong in C? What's wrong in C? XO condition in human as found in Turner syndrome. Till here correct. Turner syndrome correct. 44 plus XO, that means X0. There is no one chromosome, it's called monosomy, allosomal monosomy. Fine. But there are determine the female. No, it's not determine the female. Leave it. You got my point? Look at the last one. Homozygous sex chromosome XS produce male in the Drosophila. My dear, Drosophila XX and XY type of one. So male fellow is what? XY, not XX. Not XX. You got my point? So the correct answer is what? Option B. This is a repeated, my dear, PYQ. Remember, it's a PYQ. You got my point? So thoroughly sex, male, female, bird sex determination, ZZ, ZW, properly prepare and go. Next question, sir.
Yes. So next question. When a cross is made between tall plant with yellow seeds and a tall plant with green seeds, so already the genotype is given, so you should be knowing how to take the gametes and all. What is true regarding the proportion of phenotype of the offsprings in F1 generation? So you should know how to take it. So this one I am going to take as the male gamete. So how many gametes they can form? They can form 4 gametes. Capital T, capital Y, capital T, small t, capital Y, and small t, small y. Like this, 4 gametes are actually possible from the male. Then the next one, there is tall plant with green seed, that is capital T, small y, small y. 2 female gametes only they can form. So what are the 2 female gametes?
Capital T, small y, capital T, small y.
Small t, small y. Small t, small y. Now let's see what are the phenotype and genotype. So here first box, sir, what you think the first box is?
Capital T, capital Y, small y. Yes, tall and yellow.
Tall and yellow. Then this one,
capital T, small y. So tall and green.
Tall and green.
Come on, comment, students.
Yes, keep commenting. If you are getting, keep solving. And if you are getting the answer, please comment. So this is also tall yellow. Then next one you can see it can be capital T small t and small y. That is tall
Yellow.
tall green.
Tall green.
Then next one that is capital T small t capital Y small y, that is also tall
Yellow.
Then this one capital T small y small y, that is tall
Green.
Then here small t capital Y small y, that is dwarf yellow.
Yes.
Then last one small y
All are
small y,
recessive.
that is dwarf green.
Find out.
So it is yellow is dominant, green is recessive. Now let's see what is the ratio. The question is how many tall green are present. So number of tall green, if I am taking, you can see 1, 2, 3. So 3 tall green are actually coming. So no need to check the other one. Dark green only 1 is coming. So 3 out of 8 is tall green and 1 out of this 8 is dark green. Hope
Yes.
Okay,
Got my point?
Completed. So that's all. Like this application question also you can expect. Please keep on doing it. Come on, coming to the next one.
Look at here. Next again. Very important question. Look at here, all of you, what it is given. The question tells us, my dear, Down syndrome is caused by an extra copy of chromosome number 21. 21 is autosome. 21 is a trisomy. Yes or no? Trisomy. That means instead of 2 homologous, 1 more chromosome come and it become a trisomy. Fine. What percentage of offsprings produced by an affected mother and a normal father? Wait a moment. All of you students try to think this question. Down syndrome women, 21st chromosome trisomy can be fertile beta. She will produce 2 type of eggs. How 2 type of eggs? Look at here. I'm writing 45 chromosomes plus XX. I'm writing her karyotype. Who is karyotype, my dear? Down syndrome female karyotype 45 plus XX. I'm writing, my dear. Normal female karyotype is what? 44 plus what, beta? XX. 44 autosome. That means 22 pairs of autosome and 1 pair of sex chromosome. But in case of Down syndrome woman, 45, my dear. Because 1 extra chromosome, autosomal extra, so it's trisomy. Now tell me cross way. 44 plus XY. This fellow is normal fellow. So tell me she will produce 2 type of X. One is 23 autosomes plus X chromosome. We know in meiosis division chromosome number will be half. But look at here, 45, 23 autosome plus 1 X chromosome, sex chromosome. What about the other egg? Can any one of you guess? 22 plus X chromosome. 22 plus X chromosome. So this is a normal egg. This is a trisomy, abnormal, a non-disjunction chromosomal egg. What about this male? Can any one of you tell me male person produce a normal sperms beta? 22 plus X chromosomal sperm. 22 plus Y chromosomal of sperm. I hope you got my point. If this normal egg is fertile with normal, 22 plus 22, 44 plus XX. Normal baby. What about this one? This one is fertile with this sperm. 23 plus 22, 45 plus XY or XZ. Sorry, what beta? It is a 50% chance of this both baby. So they're asking what percentage of Turner what they call Down syndrome babies. What is the answer? What is the, how simple, my dear? Look at here. The answer is what? Who is that? Kavya. Very good.
So
The
50%. First
yes.
comment was by Kavya.
So this type you have to cross it. Turner syndrome, females are sterile. Turner syndrome, Turner syndrome, there is no X chromosome. 1 X chromosome is not there. Don't get confused. Klinefelter also sterile. Klinefelter is a trisomy of sex chromosome beta. Turner syndrome is monosomy of sex chromosome. Don't get confused. Who is this? What is this? Tell me. Down syndrome is autosomal trisomy. Remember, you got my point. Trisomy. So answer is what? 50%. Yes. Next question. Look at the next question. Come, sir.
Already this question.
Oh, very simple. Come on, try to identify this one.
A chromosome is having heterozygous condition at 4 locus. Let me put 4 locus A, B, C, D.
Position of gene.
So position of gene in a chromosome is called locus. So at every locus, heterozygous condition is coming in a pair of chromosome means what can be the number of gametes? Same equation that was said by sir, you can use it.
How many heterozygous, my dear? 4. They get 4, right? Diploid organism who is heterozygous for 4, let's say 2 power of n. I think many students are from math background students. Come on, beta, fast.
Already Muhammad
Very
Hisham
good.
has
Isham is very
commented.
good. So answer is what? Tell me. 2 power of n. N equal to what, beta?
Okay,
Number of
first comment
heterozygous.
by again Kavya has commented.
Yes, good. Very good,
Yes, the
Kavya.
answer is 16.
Right? 16
2
different
to the
type
power
of
4, you will be getting 16. 16
Type
type
of
of gametes are
gametes.
possible.
Next question. Next question. Look at the next question. One of the parent of cross this you have to be very cross. This is a HOT question, my dear, and repeated question. This is, can any one of you answer this question? Try, think, peacefully think, but this is a little advanced question. You should have a good deep knowledge in the genetics and also human reproduction. What is given? One of the parent of a cross has a mutation in its mitochondria. Does mitochondria have DNA? Does mitochondria see? Yes, sir. It is a what? Mitochondrial DNA we discussed in the last class. Chloroplast also, semi-autonomous organelles in 11th grade we discussed. Mitochondria also have. Nebenkin. In that cross, that parent is taken as a male. It is a male, right? Let's take here. This is secondary oocyte. Secondary oocyte, haploidal egg. This is entirely 23 chromosomal DNA is there. This is called ooplasm. Fine. This is secondary oocyte. Now this is sperm. Sperm is coming. Look at here. This is sperm. Head part and also acrosome is there. This is haploidal DNA is there. And this is the tail part. And this is what? Middle piece. In the middle piece, lot of mitochondria are there, beta. We know. We discussed already. This is here exactly this area where mitochondria are present. In this mitochondria, DNA is there. But this DNA got mutant, beta. This DNA got mutant. You got my point? Fine. Now, male, during segregation in F2 progeny, the mutation is found in, where is the mutation found? This mutation is going to be found in where? 1/3 of the progeny, none of the progeny, all the type of progeny, 50% of the progeny. Any one of you?
Already
Why?
commented, sir.
Why?
Kavya,
Can anyone of?
by Kavya.
Why? Tell me, any one of you? Why? Why
We
it is?
need the explanation. What is the reason?
What is the
Answer
reason?
all are commenting.
Yes.
It is none of the progeny. So
Yes.
what is the reason?
What is the reason?
What
Can
is the
anyone?
term for that? Which type of inheritance?
Inheritance
it
is a simple,
Which
beta.
type
Simple.
of inheritance it is?
Come on. Because at the time of conception, fertilization, what will come into this, my dear? Haploidal nucleus only will go, right?
Only contribution.
Only
from the
contribution
father.
is what? Father is what? Only haploid nucleus, 23 chromosome will not come. What will not come? Mitochondria DNA will not come. Mitochondrial DNA will not go inside. They will not participate in the what?
Sir? Keep telling.
Tell me here. The answer is what? None of the progeny,
That
my
everyone
dear.
commented. We need that
Ah.
term. It is an example of which inheritance?
It is okay. Then what is exactly inheritance? Cytoplasmic inheritance. Wait a
example
minute.
of cytoplasmic inheritance.
One minute. Can any one of you happen if it is same thing? If I ask a problem in the female mitochondria, here it is. I'm writing mother mitochondria or female mitochondrial mutation. Tell me the answer. Who will affect? Tell me the answer. Who will affect? Yes, good, my dear. Yes, good. Magashri, correct, beta. Diya. Good, absolutely. If it is a problem, any one of, yes, that's so tell me what mitochondrial DNA you and I receive, beta? Mother. It is called maternal inheritance, cytoplasmic inheritance. Very good, absolutely. I love you, beta. Very good. Come to the next question.
Now, the law of independent assortment can be explained with the help of. Actually, from monohybrid experiment that was conducted by Gregor Johann Mendel, he explained two laws. First law is law of dominance and the second is law of segregation. According to the law of dominance, you should understand, for every character there are two factors, one from father, one from mother. And in F1 generation, one is expressed and the other is suppressed. The one which is expressed is dominant, one which is suppressed is recessive. And even the law of segregation, which is the universal law, was explained from the monohybrid experiment. In that you should understand, genes or factors will segregate during gamete formation. But law of independent assortment, the formation of new combination or recombination, that was actually explained from the dihybrid cross. So from dihybrid experiment only, Mendel was able to explain the law of independent assortment. Now moving to the next question.
Look at here, beta. Which three scientists independently rediscovered Mendel's law? My dear, Mendel law rediscovered, nobody considered. Do you remember when we discussed the theory, when Mendel was identifying these laws and giving these ratios, mathematicians were not understanding. There was no much media, no printing, and moreover everyone was busy in where Darwin theories. Lot of what they call reasons are there which ignore Mendel, what they call laws. But after Mendel's death, tell me three scientists. One is called, tell me, Sutton. No. Tell me here, Hugo de Vries, Corners, and also Stenmark. Look at here. When I say this, Avery, MacLeod, McCarthy, something you should be remember. Griffith experiment, right? You got remember Griffith experiment, right? Again, he conducted from the dead mice and he took that DNA removed, proteins removed, RNA removed, right? But here tell me, De Vries, Hugo de Vries, Corners, and also Stenmark. But wait a moment. Who is this fellow? Did you recognize this fellow, my dear, in evolution chapter?
Yes. I want you to comment. From where you know about this person?
Hugo de Vries. Tell me. Hugo de Vries is Sabas, very good. Random and what? Directionless. Paisa. Random, directionless, and what? Tell me. It is called a discontinuous variation.
Very good. Mutation theory was also proposed by
Hugo de Vries.
Please remember the place. Hugo de Vries is from Holland, Karl Correns from Germany, and Erich von Schermach from Austria. These three scientists only rediscovered Gregor Johannes Mendel work after in 1900.
And students, I have a doubt. He used one more term in evolution chapter. Can any one of you tell me? He used one specific term in this mutation. He used one specific term.
Large mutation.
Mutation. Something.
Please comment. Single step large mutation.
He given one something specific name, term to that one. Any one of you? Very good. Who is this? Very good. Absolutely. Next question, sir. Now I'll take it this. Look at here. In fruit fly, dumpy wing are shortened and broader than normal wing. The alleles for normal wing is D, capital D. Dominant. This is a dominant one. To the dumpy wing, what is that? Allele, dumpy wing of small d. Small d is given. It is a recessive one. Two normal winged flies were mated. What? Two normal. Take capital D, and also capital D, you will get 0. You will get 0 in the mark. Produce 300 normal wing and 100 dumpy wing. That is a 3:1. 3:1 ratio. So when we will get 3:1 ratio, if you take both parents heterozygous, capital D small d cross with capital D small d. Simple, my dear. Tell me, capital D, small d, capital D small d, small d, 75%, 3:1, right? 3:1. So they're asking what a parent, what they call probability. What is the genotype of parents? How simple. Come on, answer. Very good. What is the correct answer of this one? Now tell me. Is this one? No. Is this one? No. Is this one? Or which one is the correct answer? Tell me. The right answer is option C. How simple question. Any one of you?
So anyone? Yes. Capital D, small d.
Yes. Okay, capital D. So small d, right? Capital D, small d. This is capital D. You got my point, right, beta? Capital D, small d.
Capital D, small d. Double heterozygous when they are actually crossing. Both the
heterozygous condition.
It will surely give 3:1 ratio.
1 ratio. The 300 given. 300 nothing but what? 75%. And one is what? It is 100 is what they call 7. Next 100%. No, 25%.
Next question. Chromosome maps or genetic maps were first prepared by. This was also a NEET question coming in 2019, and last year also this was actually repeated. The scientist actually who prepared genetic maps. That means locating the position of genes inside the chromosome, like body color as well as the
wing,
eye color as well as the wing shape, like that. The location of genes inside the chromosome using the percentage of recombination as the distance between the genes.
Linkage genes, we discussed this.
This was actually first discovered by Alfred Stewart Event in 1911. So this is a previous year question. Make sure you are getting it. Don't actually choose the one like Morgan and all. Morgan only said about linkage, recombination, etc. But that ideology has been expanded by Alfred Stewart event.
Remember, this time you should properly prepare the recombination and also what linkage, my dear. You have to cross check that one. Where is the genes are close? If genes are close, what? Linkage genes. When linkage genes are what, beta? They will not segregate. So more parental characteristic will come and recombinations is less. Keep in mind.
This also.
Right?
So you know about the body color and eye color. If you are taking, you can see they show very less recombination.
Yes.
1.3%. That means they are located very close to each other.
Linkage genes.
We call it as which gene? Tightly linked genes. But
Remember,
here you can see 37.5% is the recombination distance is very high. That means they are called which linked genes? They are called loosely linked genes.
Cross A, cross B is there, right?
Yeah.
In this one
you have to remember this ideology only has been used by Alfred Stewart event. Next question, sir.
Next question. Look at here. The same related question. Can any one of you answer this? Same. Just now only sir explained it here. The distance between the genes are going to be determine the recombination. But the given here, this is one of simple question, beta. If you have a knowledge on this recombination linkage, you can easily answer. There are 3 genes, small a, small b, small c, right? Percentage of crossing over between A and B, how much, sir? It is 20%. Fine.
B and C,
B and C is what? 28%. Next what? AC, right? AC is what, tell me? 8%. Which genes are linkage genes, my dear? Which genes are close? Good, very good. So I can write what, tell me? AB is what, tell me? What they call AB between these two, how much it is? The AB
20%.
20% is given, and AC
8%.
8% and BC 28%. So answer is what, tell me? Option A. Am I correct? Yes, keep in mind.
Now next one, the distance between gene is measured by. So you should remember it is actually measured using map unit. Alfred Stewart started using map units of centimorgan. 1 map unit or 1 centimorgan means 1% of recombination, like sir was telling. So when you are drawing the chromosome in that BAC, just now we have done, it is how much, sir?
20%. 20 units like
8%. That means 8% is the recombination, and the distance between them is 8 map units. And between B and C it is how much? 28%. Like that, you should take the percentage of recombination as the distance between the gene, and the unit for measuring that is map unit or centimorgan. 1 map unit or 1 centimorgan means, understand, the percentage of recombination is 1. So that is also a previous year question, check 2020 as well as 2019 question paper. Even the last year, you can find this question in the recent years.
Very easy, beta. Just cross check the distance. If genes are close, that means linkage. You got my point. And read the carefully. This question can come for case studies. Okay, case study question. Look at here. This question is you have to cross check properly. The alleles found in haploid organism cannot be dominant or recessive. Why? Alleles, we know. If you able to recall cell cycle and cell division chapter in 11th grade, alleles are located on homologous chromosome. If I take these 2 homologous chromosome, for example, here I am taking capital A, here it is taking small a, homologous chromosome. Tell me if any one of you have an idea about the 11th grade, which stage of meiosis these 2 homologous chromosomes are going to be segregate? Any one of you? Meiosis 1, anaphase 1. In anaphase 1, meiosis 1, capital A separate, small a separate. That means in a gamete, 1 single allele, beta. 1 single allele is coming. So how can 1 single will be dominant and recessive? Yes or no? If when both alleles present, only right, it is a dominant or recessive will present, right? So look at here. Dominance and recessiveness is described interaction between 2 alleles of the same genes in the same individual. Because only 1 allele is present, allele is haploid, organisms are always dominant. What about this condition? Got my point? Now leave it. Now alleles are haploid, individuals are transmitted like mitochondria. Leave it this one. No mitochondrial allele here. Most haploid individuals are bacteria and also bacterial genotype. No beta, what is the correct? Absolutely. The correct answer is what? Tell me. Answer A. Remember, haploid cells are there. Haploid cells are gametes. Gametes have a 1 out of 2 allele, right? Next.
Next question, already we have done this question, so I will be just telling the answer. We have given the distance between the two genes inside the chromosome, so just we have to take P and Q, it is 4 units, and P and R, it is 11 units, and you can see Q and R, it is 7 units, 7 map unit it is coming. So we just have to arrange them inside the chromosome.
Simple.
So P and Q, 4 we can take, and Q and P and R, how much it is coming? 11
yes,
it will be
good,
coming,
good.
and here it is around 7. Q and R, you can see it is 7. So the answer is C.
Absolutely, very good. Very good.
Like this application question you can expect from this topic. So please note that the percentage of recombination is the distance between the gene, and you have to identify the location of the genes using it. It was proposed by Alfred Stewart.
I answered that. This is not correct, better. You just cross check properly. It's not correct. It's a given what? Tell me 4 units.
No,
right?
you have to check the length
this
also.
one. Look at this one. PQ.
Length you should take.
PQ is 4
Based
beta.
on that only. Yeah.
So you have to just cross check. Where is the distance? If it is less units is there, that means the close. How simple. Next. Next. Any one of you can you give me this answer of this question? Look at here. What is the probability that a cross between true breeding pea plant, what there are true breeding with smooth seeds and true breeding pea plant with wrinkle seed will be produced in F1 generation with smooth seed? They're asking. Any one of you? Yes, sir, that good better. Come on, try to do this one. They're given true breeding pea plant, smooth seeds, true breeding pea plant, wrinkle. So here it is, wrinkle we know recessive. So if I take this cross, capital S, or you can capital R, also capital R, and also small r. In the next generation, all will be what, my dear? Smooth seeds will come, right? So answer, absolutely. So answer will come what? The wrinkle will be not there. So answer is what will be? Answer is 1. 1 will come. How simple. Next
question. Given diagram shows a pair of homologous chromosome during meiosis. Maximum crossover will occur between the genes. So 2 chromosomes are there. Let's take this as first chromosome, this as second chromosome. So you should understand maximum crossover will be taking place between 2 genes. So here you can see, here you will be getting 1. This 2 will get exchanged, and this 2 will get exchange.
Yes. Very far. Look at the distance. Look at
so
beta.
When they are very distant from each other, there is maximum chance of recombination or new combination.
Crossing over.
So you will get which combination? A will be coming after getting exchange. Here what will come? Small d. And here what
capital
will come?
D.
Capital D. So you can get a good number of which one? Sir, that one won't change.
Yes.
Only one will change.
Yes.
Either this one
or this one. Linkage.
Change.
Crossing over between the non-homologous chromosome.
A with small d and small a with capital D. Like that you will get maximum crossover. So the answer is this one. Hope you got it. Yes or no?
This one, student, keep in mind, beta. When distance is more, crossing overs takes place between the 2 non-sister chromatids of homologous chromosome can make you the new recombinant. That means new characteristic feature. Here what happened? Tell me. The genes will be what? Tell me, far. So which all genes are far? A, D are far. So answer is what? Tell me. Yes. Good.
That also.
Good. Next question.
Hope you got it. These are actually sister chromatid. These 2 are
non-sister chromatids
of
homologous chromosome. Done?
Between that only exchange will be taking place. Next.
Look at here. Given pedigree shows the trait is inherited as a autosomal dominant. What is given? Autosomal dominant. Tell me, my dear, autosomal dominant condition will be expressed in this condition also. Autosomal dominant condition expressed in this condition. That means homozygous condition, heterozygous condition. Both condition it will be expressed. So both condition it will express. Any one of you give me the correct answer of this one. They are given. What they asked? Trait the genotype of a mother and father. These 2 they are asking. Now here, if mother is capital A condition, all would affect. All would affect, right? So here who is she? Tell me, capital A small a. Here it is small a small a.
Oh, no.
This fellow is what? Tell me, small a small a. How many of you given? The correct answer is what? Tell me now here. Heterozygous. Father is what? Tell me, small a small a. Mother is capital A small a.
Yes, Kavya has given, sir. Kavya has given already,
correct?
already.
Yes, good. You got my point.
Yeah.
Right, next.
Next one, study the pedigree chart of family showing inheritance of sickle cell anemia. You don't have to check the pedigree chart. You can tell which trait it is directly. Can you please comment? Sickle cell anemia is an example of autosomal recessive disorder.
How simple.
Okay, that's simple only. So we are not going to check with that. We shall move.
Now look at here. This careful look at here. It's very easy. I hope I completely trust all my students. Can any one of you answer this? Sickle cell anemia, glutamic acid. Glutamic acid, 6th position, right beta? And hey, where? Beta. Next, glutamic acid is replaced by what? Tell me. Valine. So answer is what? What is the correct answer of this question? Fast. Ridya. Good. Very good. Very good. Very easy. GA. No sir. What is this? Tell me. GUG. So answer is what? Option D.
So please remember, it's an example of point mutation where you can see the GAG will be mutated to GUG. So instead of glutamic acid at the 6th position of the beta chain of hemoglobin, it will be replaced with valine. Now next one.
Look at here.
You can.
Look at here. Read this question and try to answer this. Which one is the correct? In prokaryotes, single type of RNA polymerase can transcribe mRNA, tRNA, rRNA. Correct or not?
That
Correct
there
or
is
not?
no separate RNA polymerase 1, 2 and 3.
Because this is what? What is this? Moniran, prokaryotes. So I can write what beta? Correct statement. In eukaryotes, RNA polymerase 1 transcribe rRNA 20S and 18S.
So in case of eukaryotes, you should understand they have separate RNA polymerase 1, 2, 3 that will be actually forming. Polymerase 1 will be forming.
Polymerase, no, I told you in the eukaryotes only polymerase 1, 2, 3 are there beta. In case of prokaryotes, one single RNA polymerase enzyme have ability to do transcribe all type of RNAs. Now look at here. This one also correct. RNA polymerase 2 transcribe hnRNA in eukaryotes, correct. And ribosomal large subunit P and A sites. So what is the correct answer of this one? Yes, absolutely all are. Yes, correct. So I'm going with what option?
So please
Yes.
remember, RNA polymerase 1 will be forming rRNA, RNA polymerase 2 will be forming hnRNA that will undergo splicing, capping and tailing
Yes.
in order to form the functional mRNA, and RNA polymerase 3 you can see it will form tRNA, snRNA and all.
Tell me here, you remember my dear, this is many times I explained here, rRNA is what? Abundant and also larger RNA. Can you look at this question? Very easy. Come on, sir.
So the disease sickle cell anemia is caused by the substitution of, already we have done, sir has already done the question. Here you can see which one is getting replaced. Glutamic acid will be getting replaced by valine, and it is getting replaced at which position? It is getting replaced at 6th position of beta globin,
yes,
chain of hemoglobin. So in the beta chain, you know that the hemoglobin will be having 4 subunits. In that 2 will be alpha and 2 will be beta.
Good.
In the beta chain only, the 6th amino acid is getting mutated because of a point mutation. So we are going to the next question. So which is the answer, sir? Glutamic acid replaced by
valine, 6th position and
valine,
beta.
6th, yes. B is
Next.
the answer. Hope you got it. We are moving to the next one.
Look at here. Operator site, promoter site, structural gene and regulatory gene the given. Any one of you? Can any one of you give this question? Molecular base of inheritance, operator site. So into the operator site, what will come and bind? Now I'm writing here it is operator site is list of promoter site, structural gene is given and also regulatory gene is given. I can write operator gene is binding of repressor protein is coming and binding to what? Tell me, operator the blocking, right? Binding of repressor molecule. Promoter site is what? Tell me. RNA polymerase. Binding of RNA polymerase. Structural gene code for what? Protein. Regulatory gene code for what? Repressor. Am I correct? Correct. Fast. Tell me. So the right answer is what, my dear? 2, 1, 3, 4. Where is 2, 1, 3, 4, sir? 2, 1, 3, 4. Right. 2, 1, 3, 4. Option A. Am I clear? 2, 1, 3, 4. Correct. Right.
Yes.
Go to the next.
All are commenting, sir. Fast. So next one. An individual is affected by phenol ketone urea lax enzyme that converts the amino acid. So here you should remember, in phenyl ketone urea there is the chromosome which is having capital P, which will produce an enzyme, phenylalanine hydroxylase. So phenylalanine hydroxylase is the enzyme that is actually produced by this gene. So it will be converting the amino acid
Phenylalanine
into
tyrosine.
very good, tyrosine. Hope you got it. And tyrosine is involved in the production of what? The skin pigment melanin, as well as tyrosine only combined with iodine to form thyroxine. And adrenaline formations. Like that, many hormones production, many character production is based on this capital P. So if small p is coming, what happened? This enzyme will not be produced. Phenylalanine will start accumulating in the body, leading to multiple effects. So phenyl ketone urea. Here there is mental retardation and here you should understand it's an example of pleiotropy, multiple effects by a single gene.
One mutant gene can give many effects, keep in mind. So it is phenylketonuria, phenylalanine and tyrosine.
Yes, that's all.
Answer is right. Look at the next question. Lactose operon induced enzymes. Lactose operon. How many structural genes? 3, right? What are they? Z, Y, A. Find out the right answer, will give chance, sir. Any one of you fast.
Let's
tell
see
me.
who is going to come in fast. Yes, Faisa.
Good. Faisa is will try rock anything. Good, very good.
Many are coming, sir. It's
beta galactosidase enzyme. Next permease enzyme and also trans-acetylase. Am I clear? Done. The order is correct. Yes. Next.
Next question. Which of the following is mismatch pair of disease and its related symptoms? Phenyl ketone urea, they have written urine turns black on exposure to air. This is another disorder which is not there in NCERT. Alkaptonuria. In that disorder only you can see the urine will be having alkaptone or homogenesic acid that can actually turn the urine into black color. So it's not a symptom of phenyl ketone urea. Remaining you can see Down syndrome, physical and mental retardation, Klinefelter, sterile males with gynecomastia, development of breast and all, and it is the trisomy of 23rd pair with 44 autosomes and XXY. And Turner syndrome, that is an example of monosomy of the 23rd pair, 44 autosomes plus XO. And they are also sterile. Yes or no, sir?
Right. So answer is what? Tell me. What mismatch one? In eukaryotic cell transcription, RNA splicing, what tell me? RNA splicing, RNA capping takes place inside. They are asking what? Tell me. Eukaryotes. Any one of you cross-check, sir? Cross-check that chart. Any one of you?
Who is going to come in? Let's see.
Good,
come on.
Fast. Faisa will get good marks this time. Full marks. Now tell me.
Very good.
Ribosome.
Keep on coming. Okay,
tell me everything. They also, right?
Yes. They are coming, sir.
Right. Next.
So it is nucleus. Genes regulating many phenotypic characters are called. Just now I said one gene that produce multiple effect. That is phenyl ketone urea, which is an example of pleiotropic genes.
Look at here. Carefully look at here. Read this question and try to answer this. Select the correct statements out of 4 given below about a lac operon. Fine. Lac operon. Glucose or galactose may bind with the repressor and inactivated it. Glucose will bind or lactose will bind? Low lactose will bind, right? Don't know. In the absence of lactose, the repressor bind with the operator region. Tryptophan act as an inducer for the gene expression. Regulatory gene is the one that produce the repressor molecule. Choose the correct option the given. Any one of you given, what is the correct options better? Come on fast.
Yes, sir.
Come on fast.
Sir,
correct is what?
It's correct only.
So tell me 2 and
4.
What they call 4. So answer is what?
2 and
Option
4.
is C. Good. Next.
Very good, very good. Keep commenting like that.
Very
You
good,
can
very
have a
good,
revision
very good,
also.
very good.
Now next
Ah.
one. Which of the following statement about Griffith's experiment are correct? A strain have a mucus coat. You should understand the strain of the Streptococcus pneumoniae or Diplococcus pneumoniae that was virulent. It was covered by a smooth polysaccharide coat. That is correct, no sir?
Yes, yes. This is what tell me virulent, pathogenic. Next.
Pathogenic. They can cause the disease. Second one. A strains are virulent is cause pneumonia infection while R strain does not.
Yes.
So understand the Diplococcus pneumoniae which is having the smooth polysaccharide coat which is getting into the body, they can cause pneumonia, whereas the other one that which is not having the smooth polysaccharide coat, that is the R strain which is not virulent. Now next one.
Transmission principle is associated with the genetic material of R strain. Is
It's
it
wrong.
R strain or S strain? Is it R strain or S strain?
It's S strain.
Now, transmission of R strain into S strain can takes place into a test tube. Can you, any one of you answer? Correct answer, my dear. Come on, come on, come on. What is the right answer? How many statements are given? Which of the following statements are correct is given, right? Now, this is the correct one. This is also correct one. And here, transmission of principle is what tell me? This is genetic material of what tell me? S strain. You got my point? S strain. So correct answer is yes. Good. Very good. So answer is what, my dear?
So remember transformation. R strain will be uptaking the DNA from heat killed S strain and they will be producing the smooth polysaccharide coat.
So they
changing
become
into
yes.
S strain. That is called transformation.
Yes. So final answer is what, tell me? 1, 2, and also what, tell me? Fourth one also, right?
Yes.
Yes. So answer is what?
So this can happen in a test tube. So
Yeah.
this was experimentally
In vitro.
proven. Who actually experimentally proven the material that they are uptaking is DNA, not the RNA or proteins? Can you please comment?
Yes,
I think
yes,
sir
yes.
already said about it in the previous
In the
question.
previous MacLeod, do you remember this one? Yes. Good. The next question.
Okay. So please comment which one? Now, what is
Very easy.
true about determination in chickens already?
Come on, come on, come on. Fast, fast. This one. Yes.
Yes.
So what about this one?
Kavya has
What
commented
about the
first by Mohammed
true
Hisham.
sex determination in chickens? That means birds. Fast. Very easy. Very, very easy.
Which one?
Fast, fast, fast.
Two set of autosomes plus
Yes, yes, yes.
ZW
Very
that
good,
is
very
coming
good.
in
Who is this?
female,
Dia.
and you can see two set of autosomes plus ZZ that is coming in males, and
Just-
these females can produce two type of gametes. So here it is female heterogamety, autosomes plus
So
autosomes plus Z or autosomes plus W, but here it is homogamety, autosomes plus Z. So female heterogamety is actually observed in chickens and all. So the answer is 1.
Joseph, it's not correct. But a cross check. It is not XXXO. It is in grasshopper, my dear. It is ZZZW.
We
Okay.
cannot consider X here. We consider it as Z and W only.
Next.
Can you read? Which of the following represent the correct order of genes on the chromosome? Same thing only like Alfred Stewart Evans experiment. Here you have to arrange the genes correctly inside the chromosome. So many are coming. So you have to take the crossover value as the distance between the gene and you have to arrange them correctly. So here, if I take T and U, the distance between them is 25 map units. The distance between them is 25 map units. So let's take T and U. In that, the distance that is coming is 25 map units. And in that, T and V, it is coming as 5. So T and V, you can see it is coming as Y. And V and U, it will be coming as 30. And U and W, it will be coming as 20. V and W, it will be how much? So how we can have to take this one? Just check, sir.
The linkage here. Look at here. The linkage is unit is what? Linkage pair is what? TU is 25, right? TU is 25. TV is 5 only. That means they are very close.
Yeah.
The TV is 5 only, right? Good. And VU is what? 30. That means V and U is what? Tell me 30. That
That is
means this
also
is what?
correct.
30.
10.
And UW is what? 10 only. U and also W. So your W we can write here. It is what? U and W is 10 only, right? So here if we take here, it is tell me VW is 20 is given, right? So VW is 20. So this is not, we cannot, right?
Yeah,
Now
so let's see which one will work out.
this will be Tell me, we can write here it is TV, right? TV, WV, right? W,
TWUV. That will match, I think, TU.
5%. That means TV are close, and after
So
that,
which
tell
one
me
is matching? Just we
VU.
have to write and see.
VU is what, tell me, 20.
VTWU.
VTW will come, right? VT is what, tell me, here it is
VT it is 5.
5. Next TV.
TV.
No.
That is coming around. Which one? So
TU, actually. TU is 5. TU is 25.
yeah.
TU is 25.
Then
Next TV 5.
TV
So
is
that means
5.
TV.
I think that question.
TV is 5.
Then
TU is 25. VU is 30. VU
and you
is
should
30.
combine two and four.
And next to UW 10. UW 10 and VW is 20. VW is 20, right? This one is 20.
matching.
Yes, correct, right?
Yes.
Yes, so we can
So
go
the
with
answer
option
is
C. Shall we?
Hope you got it.
You got my, shall we go with this one? Shall we go with this one? If
So many
A,
commenting A. How?
T, VT is what, tell me, VT is how much?
VT.
VT.
It is not given. Just leave it.
Leave it.
TU.
This one is TU.
TU just check, sir.
TU is 25. TU 25. Right?
TU that is 25.
If it is, this one is 25. If it is TV, TV is what, tell me, 5. TV is 5. This is not correct. Right? Next one is called VW. VW, how much it is given? 20. VW, if it is 20. And TU. TU,
Yes,
it is.
sir. Here only it will come, sir. Then
it will
is
be
correct.
what, tell me, option?
It will be correct in the first only.
First only, yeah?
Yeah, this is
TV,
correct actually.
TV is how much?
VTWU. VTWU. TV it is 5. VW.
TV is 5. Okay.
Yeah. Then
And VW?
VW it will be 20.
VW is
VU will be
20.
30.
Okay. VU is what? 30.
30.
Right.
Yeah.
TW?
TW it will be. It's
TW is 20.
Yeah.
TW is 20. All
TW
right.
is not given, sir. So that
sir,
is only
this
given.
one? Okay,
so
okay,
with the
right.
available information, A is correct. A is correct.
A is
So here W
appropriate.
you have to take in between. That's all. Hope you got it. So it is one only.
Now look at here. In eukaryotes, transcription occurs in nucleus. That means mRNA and also what, tell me, crossing over. What? It's called what, tell me? mRNA also splicing, right? Splicing, everything takes place in nucleus. Correct. In bacteria, transcription and translation occur in cytoplasm. Yes, correct. This statement also correct. But I can go with what, tell me?
Both
option.
are correct.
Got my point? Go to the next question, sir. Last question.
So, which of the following condition can lead to erythroblastosis fetalis? This already you know. If the father is Rh positive and the mother is Rh negative, you should take here as positive. Here it should be taken as negative. So when the positive gene is coming from father and negative from mother, the first child you can see on the surface of RBC, they will be producing which antigen? They will be producing the Rh antigen. Got it? So when this first child is delivered out, only the problem will happen, because this blood will get exposed to the mother and the mother will produce Rh antibody. The mother will be producing Rh antibody. And when this Rh antibodies are produced, first child no problem, but the second child only the problem will happen whenever they are having the RBC which is having this Rh antibody. Sorry, Rh antigen. Because this antibody will come and attach to this and it start bursting the RBC, that is leading to erythroblastosis, the bursting of RBC. And it can lead to the death of the child, that is called erythroblastosis fetalis. This can happen when it passes through the placenta and attack the RBC of the child. So the genotype that you should take, the father should be Rh positive homozygous and mother should be Rh negative homozygous. So the answer is this one and this one. Both are actually matching. First child is Rh positive also, this can happen, but this will never happen. So it is both 1 and 2. Okay, many of them commented.
Look at the last question, my dear. Look at the last question. Today's last question. Very, very, very important question. Regulation of gene expression. Remember all of you what they're asking. Regulation of gene expression can be exerted in eukaryote. They're asking eukaryotes right here. Here asking eukaryote. So what is the correct answer of this question? If you read NCERT, in NCERT clearly mentioned that the eukaryotic gene expression at the several levels. What are the several levels? Transcription level, translation level, processing level. You got my point? This all levels are there. Yes, this can be come for 2 mark also. Transcription level, processing level, transport of mRNA level, and also translation level. So I can go with what? Tell me option D, all. But in the exam, if they ask prokaryotic, if they ask, what you will write? If they ask prokaryote, what is your answer? At what level they will be transcribed? Mahek, tell me what exactly the answer in case of prokaryote. In case of prokaryote, if the same question come, if you remember that lac operon, we are stopping gene expression at transcription level. That means all Z gene, Y gene, A gene stop. You got my point? So no translation, no transcription, so no mRNA formation. Remember, my dears, till here all of you go through these questions properly, practice at home. Tomorrow also we are coming with 2 new chapters, different chapters. We probably will start with the sexual reproduction in plants and human reproduction, 50 questions or 45 questions like that. This session will be 100% useful for you to get maximum marks in the final examination.
So please revise the chapters nicely, read the NCERT line by line and understand the concept. Come for our question discussion. It will be a good revision for you guys. So please join us tomorrow. So bye.
And moreover, absolutely very good, better. Today you given absolutely good answers, and that means you are practicing at home thoroughly. We have 10 days time is there. You should rock, and final examination 98, 99, 100 out of 100 only. Good night all of you. Will meet in tomorrow's class. Bye bye.
Okay then, bye.